Our research looks at how blocking androgen signaling in early pregnancy affects fetal outcomes. Female hormones, including estrogen and progesterone, are essential regulators of normal placental and fetal development. Interestingly, females also express male hormones called androgens, but their contribution to healthy pregnancy is unknown. We used a drug called enzalutamide to block androgen signaling in early pregnancy and examined how it affected pregnancy outcomes in mice. After blocking androgen signaling, we saw a striking 11% reduction in fetal weight at late pregnancy. This was associated with a 31% decrease in fetal to placental weight ratio, implying that the placentas were less efficient at supporting fetal growth. This finding indicates a significant role of maternal androgen signaling in fetal outcomes. The next important step in our research is to investigate the underlying mechanisms contributing to this observation, including how androgens could impact female immune and vascular systems, both of which play key roles in healthy fetal development.
Our research explores how the female immune system supports pregnancy and how insufficient genetic disparity between mates might contribute to subfertility. We hypothesize that male seminal fluid histocompatibility antigens uniquely shapes the female T-cell response in early pregnancy to promote implantation and reproductive success. Using genetic mouse models, combined with advanced techniques like flow cytometry and single-cell multiomics, we analyzed the T-cell repertoire and fetal development. Our findings reveal that seminal fluid histocompatibility antigens play a critical and previously unappreciated role in driving maternal immune tolerance to facilitate optimal placentation and fetal development. Our research offers a new prospect of improving pregnancy outcomes through development of tools or drugs to improve fertility both in agriculture and conservation.
Our research looks at how paternal programming affects embryo development and health. Did you know that sperm carry more than just DNA? One key player is microRNA 30A or MER30A. Its abundance is modified by environmental and lifestyle factors. But how does this affect embryo development? To investigate this, we generated a novel mouse model overexpressing sperm-specific MER30A and utilized a MER30A depleted embryo model. Across both models, we observed delays in pre-implantation embryo developmental time points and reduced blastocyst cell numbers. As a result, our research suggests that sperm MER30A is crucial for early embryo development and health. Future studies could explore screening or manipulating sperm for optimal MER30A levels to improve embryo quality in couples struggling with male-related infertility.
Our research has led to the development of a novel diagnostic tool to determine embryo health. Hi, I'm an egg specialist, not the kind you'll find in a supermarket, but the kind that will turn into an embryo and ultimately a baby. Did you know that for 1 in 6 individuals worldwide, starting a family remains a distant dream? For them, the joy of hearing their baby's first cry feels heartbreakingly out of reach, even with the help of IVF. We have developed a groundbreaking diagnostic tool that taps into a natural phenomenon, light emitted by embryos, much like how fireflies communicate in nature. By shining gentle doses of light on an embryo, our technology captures light that bounces back from the embryo and determines how this is reflective of the embryo health. This innovative technology has the potential to significantly improve the chance of IVF success and, importantly, patients finally hearing that first precious cry.
Our research focuses on how the levels of certain cells early in pregnancy might predict and contribute to the risk of preterm birth. Regulatory T cells, or Treg cells, are a specialised subset of immunomodulatory T cell that limit inflammation. If someone has few or low-functioning Treg cells in late pregnancy, it's associated with spontaneous preterm labour, and we're now studying when these Treg cell deficiencies arise. Using flow cytometry, we assessed peripheral blood mononuclear cells isolated in early and mid-pregnancy as part of the ORIP trial. Compared to women with uncomplicated term deliveries, we found that women who later delivered preterm had fewer peripheral Treg cells in early gestation. This data suggests that an impaired Treg cell response in early gestation precedes preterm delivery. We're now assessing T cell populations alongside pro-inflammatory and pro-tolerogenic cytokines in plasma to develop screening tools to identify women at risk.
Our research focuses on the crucial role of the immune environment in early pregnancy in protecting the long-term health of offspring. The health of future generations begins to shape even before birth. Our research highlights the critical role of a specific immune cell, known as a regulatory T cell, during early pregnancy. These cells maintain a balanced immune environment in the mother, which is vital for the long-term cardiac and metabolic health of the baby. Our study in mice found that when Treg cells are lacking, which is common in pregnancy complications like preeclampsia and preterm birth, young adult male offspring develop issues like poor glucose tolerance and cardiac dysfunction. Interventions aimed at preventing Treg cell deficiency during pregnancy can avert these adverse outcomes. This suggests that supporting a mother's immune system during early pregnancy could be key to protecting the future health of our children.
Our research seeks to explore how the female immune system evaluates the compatibility of a male partner. Genetic differences between parents can improve reproductive outcomes and have significant implications for human reproductive health, livestock animal industry, and endangered species conservation. Our research aims to investigate the role of the female immune system in assessing the compatibility of the male partner. In particular, we are interested in how maternal immune cells, called T cells, can sense and respond to molecules called histocompatibility antigens that are inherited from the father and expressed on placental cells. My PhD study aimed to identify the most important molecules that influence the female immune response and define how maternity cells contribute to this process. Findings from this project will help us understand how the female can selectively invest reproductive resources into pregnancy and lead to improved offspring's fitness, according to these father-specific molecules. Ultimately, this will provide innovative solutions and improve reproductive success in many species where mating compatibility is a huge issue.
Our research examines the impact of semen molecules on the female immune system, pregnancy, and fetal development. For a long time, we have thought the male contribution to reproduction was just to provide the sperm for fertilization. However, we now know that molecules in semen cause changes to the female immune system, which contribute to healthy pregnancy and fetal growth. My research aims to identify factors from the father involved in driving these changes, and one molecule of interest is microRNA 146A. Surprisingly, removing this molecule from male mice led to increased embryo implantation rate, showing microRNA 146A is a signal to the female immune response to limit the number of implanting embryos. These findings further demonstrate that male factors can both stimulate and inhibit female fertility, which play a key role in conception and pregnancy progression. More knowledge of how this works will improve our understanding of factors contributing to normal fertility.
Our research focused on IVF and how repeated freeze-thaw cycles affect embryo survival. During an IVF cycle, embryos are frozen and kept in storage while waiting for genetic testing results. When genetic testing fails to yield reliable results, embryos can undergo multiple rounds of testing and freezing. Research has found that repeated genetic testing impacts live birth rates, but the specific impact of repeated freeze-thaw cycles hadn't been fully explored before our study. In a preclinical model, we found that embryos that underwent multiple freeze-thaw cycles had a lower chance of survival and implantation and can lead to smaller, less healthy offspring. Our research is the first to show how harmful repeated freezing can be, and we hope it will help improve IVF practice to give families a better chance at having a healthy baby.